Thanks everyone for joining us for today's webinar, Morphology-Driven High-Plex Spatial Analysis of Tissue Microenvironments, presented by Dr. Douglas Hinerfeld. Dr. Hinerfeld is the Director of Product Applications at NanoString Technologies, responsible for the development of novel applications for their 3D biology portfolio. He received a BA in Psychology from the University of Colorado Boulder and his Ph.D. in Genetics and Molecular Biology from Emory University. With that, I'll now hand everything over to Dr. Hinerfeld to begin the presentation.
All right, thank you very much. So today I'm going to talk about morphology-driven high-plex spatial analysis of the tissue microenvironment. So NanoString is a Seattle-based company. We've really been founded, and our core technology has been the nCounter technology, which is a technology that's used primarily, historically, to analyze RNA from clinical samples and other types of samples. So it's a direct analysis tool that doesn't require any amplification of the RNA prior to analysis. And this technology has generated to date over 2,700 publications. So it's a very well-established analytical platform. And I'm going to talk about how we've taken that core technology and expanded it to do high-plex profiling of both RNA and protein while keeping the spatial context in clinical FFPE samples. So going back to our core technology, this was a paper published a couple of years ago by Merck. And the goal of this study was to identify a signature, an RNA signature, that could predict whether or not patients respond to their checkpoint inhibitor, Keytruda. So for this study, they took the tumor specimen, FFPE samples, grind it up, purify RNA, and analyze it on the nCounter. And they started with a larger set of genes that they thought would play a role in checkpoint response, and ultimately narrowed that down to a set of 18 genes, which they call the T-cell inflamed signature, or what we call the tumor inflammation signature. And what this particular graph shows is on the y-axis, we have progression-free survival. How long the patients survive after treatment. On the x-axis, we have their inflammation. So what you see is most patients that have good survival also have inflamed tumors measured by this bulk tumor inflammation signature. And so that's in this box here. But at the same time, you see there's a lot of tumors that are inflamed yet don't respond to the checkpoint. So it's a good indication, but there's still a lot of unexplained clinical outcome from this assay. Merck then took this a step further, and they integrated both this tumor inflammation score, what we call the TIS, and tumor mutation burden. So essentially they're asking, is the tumor inflamed, and are there large numbers of neoantigens present within the tumor based on the mutation burden that would elicit an immune response? So again, they did this in looking at their checkpoint therapy. And what this result shows, so on the x-axis is the tumor inflammation score, GAP. On the y-axis is the tumor mutation burden. So if you look at melanoma specifically, you see that 57% of the patients that have high mutation load and are inflamed, as measured by the tumor inflammation score, have good progression-free survival. So that's great. It's an improvement. But there's still 43% of the patients for which they can't explain whether or not the patients are going to respond to the checkpoint therapy or not. So in thinking about identifying these biomarkers, there's a lot of pieces that come together. So in this specific case, they're looking at PD-L1 histology, they're looking at the tumor inflammation score, they're looking at the tumor mutation burden, but yet they're still not able to really satisfactorily predict response to the checkpoint therapy. So more candidates are needed, and as you'll see as we talk through as I talk through the presentation, you'll see that spatial context becomes particularly important in this context. So comparing responders and non-responders and doing bulk analysis, as we saw with the inflammation score, it definitely has a lot of benefit. But there are cases for sure where bulk analysis does not reveal any differences between responders and non-responders. But if you can then focus in on the biology within the tumor, focus in on regions of the tumor where that biology is really active, then you can start to flesh out distinct differences that will separate the responders and non-responders. One example of why spatial context is important is in the immunoscore assay. So this is a prognostic test used primarily for colorectal cancer. And in this test, it's a low-plex assay. So they're looking at a few different immune cell types. They're looking at how many of those cells are there and, importantly, where they are. Are they on the periphery or are they infiltrated within the tumor? So they integrate the data, the types of cells, the density of cells, and the location, and that computes an immunoscore. And what the graph on the right shows is that patients, tumors with a higher immunoscore have better prognosis regarding disease-free survival and overall survival. So location matters. But in the space of immuno-oncology, it's not just location, it's also the vast numbers of cells. It's T cells and B cells and NK cells and macrophages and their activation states. So there's a lot of information that you need to gain. As we saw before, you want to do it spatially defined, but you also need high-plex analysis. And from a technical limitation perspective, there's a lot of technologies that allow you to do high-plex analysis. They're very quantitative, very precise. But when you get to spatial analysis, really the plexing is a challenge, the ability to have good quantitative results, and the precision of those technologies. So there's a lot of limitations in spatial analysis with regard to solving this biological problem. The other is what I call the pathologist dilemma, which is limited amounts of material. So you want to get all this information, you want to do high-plex analysis, you want to do spatial analysis, but you oftentimes have very limited material. So being able to do this with very limited amounts of material is an additional challenge. So one of the early studies that we did to really start to think about both bulk analysis and spatial analysis was with Dr. Aubrey Thompson at the Mayo Clinic in Jacksonville. And in this study, we're looking at the effects of the immune architecture on response to adjuvant capecitabine therapy in triple-negative breast cancer. And this trial is called the FINXX trial. So the goal is to identify biomarkers that can predict which patients are more likely to benefit from capecitabine. Better biomarkers are needed. So the question is to identify predicted biomarkers, tumors from patients that experienced either recurrent-free survival or not. So they either had good recurrent-free survival or they did not. Were analyzed using both a bulk assay that's been really useful in understanding the tumor biology called the BC360, the Breast Cancer 360 assay, as well as the GeoMx DSP platform. So the challenges they were faced coming in was, again, they didn't have enough information to predict who was going to have regression-free survival and who was not, so they needed to fill that funnel. Limited samples, so we only had 2 FFPE samples from Finland, and we did not have access to additional samples, so not really a precious small amount of material to work with. And we actually had a goal of presenting this at ASCO last year, which we did achieve. I'm happy to say was quite a bit of material we needed to get through in order to generate that data for the ASCO presentation. So there was a throughput requirement we had. So we ran the Breast Cancer 360 assay, and that was done prior to going to the GeoMx study. And that's a 776 NanoString gene panel. And this panel looks at both the tumor inflammation signature that we talked about, but also the PAM50, which is our Prosigna assay, essentially the components of our Prosigna assay, which are a prognostic assay for breast cancer. But then we have a variety of other signatures related to breast cancer biology and tumor and immune biology. So all of this is integrated into one assay, take FFP samples, extract RNA, hybridize it directly to our barcodes, and read out the strength of these various signatures. So we're not looking necessarily at just the gene by gene level, but at different signatures, as you see called out on the right. So if you look at all the samples that were run in this subset of the FINXX trial on the y-axis, and on the x-axis, you look at the expression of these different signatures, the strength of these different signatures. First off, you see a great amount of heterogeneity across the samples and across the study groups. But specifically, if you then ask about the differential expression of these signatures, or genes in some cases, between patients with longer survival on the left or shorter survival on the right, you see that those patients with longer survival, 2 things jump out. There's a strong immune component. So you see cytotoxic T cells, you see PDL2. But at the same time, these are just at significance from a p-value perspective or just below significance. So they're hints, but they're really not strong enough to sort of jump right from this into larger clinical validation studies. So this suggested that we needed to go deeper, specifically into the immune biology of the tumors in order to identify, to get more information, more resolution, better biomarkers, better candidate biomarkers. And I'll talk about how we do this with the GeoMx technology in the coming slides. But what we did was we took these tumors, we stained them for these visual markers, pan-cytokeratin for the tumor, DNA for the nuclei, and then CD45 for all lymphocytes and CD68 specifically for the macrophages. And then we selected different regions within the tumor. We segmented those so that we could look at just the tumor compartment and just the stromal compartment. And then we analyzed them with the GeoMx platform. And what we were able to see with that is if you look just at the stroma compartment in the CD45 enriched regions of the tumor, a great amount of heterogeneity. This is sort of mixing the within sample, the different regions of interest and the different patient samples. But you see a lot of heterogeneity in the CD45 enriched stroma and in the CD68 enriched stroma. So immediately you're seeing how this kind of heterogeneity could prevent you from identifying sort of really significant strong biomarkers from bulk analysis. But if you then look specifically in the stroma of the CD68 enriched regions, you start to see that patients with good regression-free survival have increases in expression of, for example, PD-L2 and IDO1, and that fibronectin is actually upregulated in patients that don't have good survival. And as it turns out, PD-L2 specifically also, as I showed before, came out of the bulk gene expression analysis. So you have some ability to cross-validate, but importantly, you're identifying additional important markers with strong significance by looking at very specific spatial regions of the tumor, as is true here with IDO1 and fibronectin. So this demonstrates, at a high level, high throughput multiplexed analysis of very limited clinical FFPE samples could reveal and then help to prioritize novel biomarker candidates. So how did we do that? So we did that on the GeoMx Digital Spatial Profiler platform. And starting at the left, this is really a combination of reagents, and then the system, essentially the microscope itself, and then data analysis tools. So in terms of reagents, we analyze either protein or RNA. Starting with protein, we use IHC-validated antibodies, and on each antibody, we tag it with a unique UV cleavable DNA barcode, shown here. We can do the same for RNA, so we take in situ hybridization probes, and we attach to each probe a unique UV cleavable DNA barcode. So those are the reagents, stain the samples with those reagents, put them into the instrument, and then we control with exquisite control where we shine the UV light. So only where we shine that UV light do these barcodes come off those analytes. And then we sip those barcodes up and we analyze them, and we can look at the spatial analysis of the expression of all the different proteins or RNAs that we're targeting in the assay. And just in a bit more detail, it's very much, at least for a protein, like a standard IHC assay. For RNA, like a standard in situ hybridization assay. In this, you mix together both antibodies that are labeled with a fluorescent marker, and you saw in the FinXX trial samples, the breast cancer samples, we use pan-cytokeratin and CD45 and CD68. So you mix those antibodies with our DNA barcoded antibodies, which are invisible at this point, stain the sample, and then that sample is ready to go into the GeoMx DSP system, which is shown here. It's an excellent fluorescent microscope, 4-channel fluorescent microscope. You get that image, then you then use that image in the morphology of that tissue to guide where in the tissue you want to analyze. So you select regions of interest. Once you've done that, you tell it to go collect the instrument, the regions of interest. It shines the UV light on those specific regions, sips off the tags, places them into an onboard microtiter plate, goes to the next region of interest, shines the light, sips off the tags, and it goes around and cycles through this until it's collected all the regions of interest on that particular slide. The system holds 4 slides, so it then immediately starts with the next slide. Once you've filled this plate with the tags that are released from the various regions of interest, those are then analyzed on the nCounter platform, or as you'll see, also will be able to be analyzed through high throughput sequencing technologies in order to count those tags and relate that back to the expression of all the markers in each spatially defined region of interest. So for the nCounter analysis, this is done using our gold standard platform, our gold standard technology, which are these optical barcodes. So we have a reporter probe and we have a capture probe. These sequences shown here in these half sites are complementary to whatever we want them to be. In this case, they're complementary to the unique UV cleavable tags that are on the antibodies or on the ISH probes. So we take those release probes, we take these barcodes that are specifically designed to count those particular molecules, we hybridize them together. Once we hybridize them, we put them onto the nCounter platform where they're imaged, and you can see these strings, these barcodes. Each of these color patterns is designed to be specific to one particular UV cleavable barcode, so we just count them. And so by counting these barcodes, we're counting those tags. And ultimately what you get is all the different analytes you're measuring, all the different samples or regions of interest, and then the digital counts, how many times that particular molecule or that particular UV cleavable tag was present in that sample. So it's a direct quantitation. So a really simple example of that is shown here, where we've stained lymphoid tissue with Ki67 and CD3. We've selected different regions of interest that are enriched based on the fluorescently labeled antibody for either Ki67 or CD3. We also have a DNA barcoded antibody for those two markers. And you can see regions that are high for Ki67 staining also have high levels of the counts from the DNA barcode. But you're also staining with all the other markers in the panel, and this is quite old data, but what it highlights is that in each of those regions of interest, you're quantitating the expression of all these different markers in the panel. So in terms of the content, the numbers of analytes that we can measure, on the protein side, the way that we've designed this content is we focus on creating a core module, and that core module has all the negative control and positive control antibodies that you need to determine background, to normalize the data. But it also has a lot of the common markers that you'd expect to see, CD45, CD3, CD8, and so forth. So that's a required core to run it, but then you can add different modules depending on the biology you're interested in. We have a pan-tumor module, an immune cell typing module, an IO drug target, immune activation status, and we continue to expand the catalog of available modules. But importantly, if you have something of interest that we don't have in our panel, which is common, you can add custom content, so you can send that antibody to us, we can put a unique barcode on it, and then you can spike into the panel, or we have a relationship with Abcam that gives you sort of more direct access to their 6,000 or so different IHC validated antibodies that they can then put barcodes on. We have a panel now for human immune profiling, human neuronal cell profiling, which is focused on neuropathology primarily, Parkinson's disease and Alzheimer's disease. And then we also have a mouse IO panel, so mouse-specific antibodies. On the RNA side, we've really just dipped our toe in the water. So we have an 84 gene panel, human IO, and again, you can add custom content to that as well. But as we get into, and I'll talk a bit more about this later, as we get into taking that plate of release tags, and instead of going to the nCounter, which provides almost immediate results, you can go into a workflow where you generate sequencing libraries, do Illumina sequencing, and then import those FASTQ files back into our software. We deal with all the demultiplexing and all the deduplicating of that data and generate essentially counts. But in this case, it allows us to expand the numbers of analytes we're measuring. So the first panel we will be releasing is about a 1,500 gene panel, and I'll tell you a little bit more about that in a bit. So one of the things that's really unique about the way that we build these panels is the level of validation that we go through. And here I'm talking specifically about the antibodies. I'm not going to go into this in great detail, but we start with IHC validated antibodies. We confirm that those antibodies work under our antigen retrieval and binding conditions. We then put the unique DNA tag on it, so we conjugate it, and then we determine that we still retain the same specificity as before we put the tag on. We then do a variety of screens for sensitivity and specificity. We make sure that those antibodies behave the same when you analyze a sample with that antibody alone, and you combine that with all the antibodies in the panel. And then finally, we have a variety of tissue microarrays that have tumor types and different tissue types that we can make sure that biologically that we're seeing a signal where we expect to and not where we don't expect to. This is just an excerpt from a poster presented at AACR by Janssen Research and Development. So they're one of the early adopters of this technology. And they did a whole variety of studies doing their own validation. This just showing some IHC comparisons with non-small cell lung cancer. So this has been validated by a variety of different groups in some of their studies, and we've seen excellent agreement in the results. So these are really well validated, off the shelf, ready to go panels of antibodies. So another interesting component of the platform is if, in that first example, I sort of showed you, round circles and squares, but then you saw in the study with FinXX that we were able to shine the light just on the tumor and just on the stroma. And the way that we can control how the light shines with such clarity is we have in the system a digital micromirror device. So this is about 1 million tiny mirrors that are each individually controlled on a chip. And when we shine the UV light on those mirrors can be directed to shine the light exactly where we want the light to shine, and only where the light shines are those tags released. So this shows a simple example of staining a core from a tissue microarray with pancytokeratin and for DNA. And then we shine the light only on the pancytokeratin positive region, and then we shine the light only on the pancytokeratin negative region. So we've taken essentially this core, or this region of interest, and optically microdissected that into the tumor in the stroma. Having the ability to control where the light shines gives us a lot of different possibilities in terms of how we can analyze samples. So we can use these basic geometric ROIs, which have been very successful. We can also use this polygon tool to draw very specific regions within the sample that you want to analyze. We can segment the sample into both the tumor and stroma, or into additional compartments based on those fluorescent markers. You can look at very specific populations. So we really have a hypothesis about a particular population of cells you want to analyze. Use a fluorescent marker to highlight those those cells that express that marker. In this case, we would shine the light just on those cells and nowhere else in the tissue. And because you have so many of these micromirrors, you can shine the light on these 3 cells all at the same time, sip off those tags. Then you can shine the light on the white cells all at the same time, sip off the tags, and then if you want, you can shine the light on everything else. So you've essentially broken that region of interest into 3 biological compartments. And then there's a variety of other tools that you can use to look at other aspects of the biology within the sample, shown here with the contour and the gridded profiling. So this is just an example of looking at FFPE cell pellets. So each one of these is a particular cell line. We've drawn just different size regions of interest from 300 micron down to 50 microns. And then we analyze each of those independently in the nCounter. And you can see, if you look at the different cell lines and you look at just the expression of CD3, if you look at it fluorescently or by the nCounter tags, you get the very same responses for CD3, CD45, and pancytokeratin. So these are the types of things that we do to validate the platform. But once you normalize the data, all of that data clusters based on the cell type. So independent of cell size, shown here, once you normalize it, or ROI size rather, once you normalize it, that data comes right together. So whether you have a 50 micron region of interest or a 300 micron region of interest, you're getting the same data. Again, with these geometric ROIs, just looking around either lymphoid tissue or tumor tissue, you really start to parse out the heterogeneity of the expression of the different markers in our panel across the different regions of interest. This approach of these simple geometric ROIs was used in 2 back-to-back studies looking at combination neoadjuvant therapy with Ipi and Nivo. One was done by Christian Blanc at the NKI in the Netherlands, and then the other was done by Dr. Jen Wargo at MD Anderson. So in these studies, they were using GeoMx to identify biomarkers that could potentially predict whether or not patients are going to respond to Ipi and Nivo combination neoadjuvant therapy. They oftentimes, based on the study design and the on-treatment sampling, had very small amounts of material, so they thought this platform would have a lot of value in that context. One of the things that was a surprise is that they actually saw that B cells were potentially predictive of enhanced response in these trials. So that was somewhat unexpected and sort of highlights the value of being able to do High-Plex analysis from very small amounts of material. Dr. Wargo has then been following that study up, digging deeper into the sort of the B cell biology and some of the other observations she made. So what she did found was that both B cells and the presence of tertiary lymphoid structures within the tumor implicated a better response. So she's following this up by going back into the DSP and further dissecting the immune content in the context of these tertiary lymphoid structures. So this is shown here where she has both baseline samples here and on-treatment samples here. And this is just confirmation by IHC that in fact the responders, particularly in these on-treatment samples, have higher amounts of CD20 than the non-responders, so more B cells, and that the tertiary lymphoid structure makes up a higher percent of the area of the tumor, or essentially more tertiary lymphoid structure in the on-treated samples, although it shows up in both, between the non-responders and the responders. So more B cells, more tertiary lymphoid structures. The next step is to dig deeper into this and understand what's going on at the biological sort of cellular level. So the way, and this is an ongoing project, this is just some early data, but what this shows here is if they look at tertiary lymphoid structures, so these are actually selected here on a serial H&E slide, and they look both within the tertiary lymphoid structure and outside of the tertiary lymphoid structure. And they've stained this sample for T cells, B cells, and then the melanocytes with PMEL and S100 beta. They then selected regions of interest both in the TLS and outside of the TLS. They then analyzed the B cells, so just the CD20 positive regions, then just the T cells, the CD3 positive regions within those, and they could look at what is the difference in the biology of the T cells inside and outside the tertiary lymphoid structure, and the B cells inside and outside. And this is just some preliminary data here, but showing if you look between the not in the TLS and in the TLS at a variety of markers in the T cell compartment, you see very specific changes. And then in the B cells, you see other specific changes. So it demonstrates really how you can use the technology to discover novel potential markers and then go back in and get deeper biological analysis of them. So another study is a project that was also presented at AACR last year by Vicky Rimkunis. She was the associate director at H3 Biomedicine, but has since moved on to Repair Therapeutics. So in this study, they combined both bulk analysis and spatial analysis, in this case, spatial analysis of both RNA and protein. And just a little background, 80% of the patients with muscle-invasive bladder cancer fail to respond to immunotherapies. This suggests existence of a complementary immune evasion mechanism within the tumors. And so there's a need for comprehensive immune profiling of patients of their tumor samples. So this is really a technical evaluation of both bulk versus spatial analysis, RNA and protein, for assessing the tumor microenvironment and defining the bladder cancer molecular subtypes. So define immune context of tumor and stroma in these, what you'll see, these PPAR gamma high samples. So what they had found is that samples that are high for PPAR gamma expression have low tumor inflammation scores. So these are these bulk analyses of tumor inflammation. So this is shown here where you have PPAR gamma high samples and they have low tumor inflammation, a score of 4.52, which is quite low. And here you have PPAR gamma low samples, which have high tumor inflammation. So there's some association between the PPAR gamma state and the tumor inflammation. And that comes from bulk analysis. They then went deeper into this, staining for pancytokeratin, CD3, and DNA, and then segmenting that tumor, that sample on each region of interest on the tumor and then the stroma. What this result shows here is that if you look between the PPAR gamma high and low, you don't see much of a difference when you look at just the stromal compartment. But when you look at the tumor compartment, you see a significant increase in the CD3 protein population within the tumor itself. So you're getting more infiltration. Just looking at that data across all the samples, so here's a variety of markers within the panel, and this is looking across the patients. You have PPAR gamma high that have no infiltration. You have PPAR gamma low that have high infiltration, like I showed, and then you have a variety of mixed samples here. And this is based on the protein analysis, and then looking into the RNA analysis between the PPAR gamma high and low using a similar tumor stroma segmentation, you start to parse out even additional markers. One interesting marker here is GATA3 shows up as highly expressed in the PPAR gamma high at the RNA level. And that's actually a gene that's been associated with subtyping of the basal and luminal. And you see that, in fact, when we categorize those samples as basal or luminal, they segregate based on GATA3. So you're able to actually get this molecular subtyping data out of the GeoMx analysis, as well as all this other context-specific, spatial context-specific information about the tumors. So shifting gears a little bit, so what I was just showing you was has been mostly protein analysis, although we hinted at the RNA analysis there in the last study. This just shows a very similar workflow, but one of the key differences here is when you're doing RNA analysis in the tissue, there's actually two approaches you can take. You can stain for fluorescently labeled antibodies, but it's quite limiting because you're only allowed to detect the most abundant antibodies under those conditions. But you can also stain with the RNAscope technology. So these are from ACD, and they allow you to fluoresce at the single cell level, or actually the single transcript level, the level of particular transcripts within a tissue. So here you're staining with both RNAscope probes for specific genes of interest and our panel of DNA barcoded ISH probes. Same thing, image it, select regions of interest based on the RNA expression, the spatial RNA expression, and then analyze the expression of the RNAs in these different regions of interest. So this just visually shows that here we're staining with RNAscope probes for RNA expression of CD19 and CD3E. We're then selecting different regions of interest based on the expression of those RNAs and profiling. In this case, this is an early panel of 78 IO RNA targets, and you see immediately the heterogeneity within the samples. If you look at these immune poor regions, these CD3 enriched regions, and these CD9 enriched regions, you see very different profiles. And of course, you can then go in and start to dissect the genes within those different regions of interest and their relative expression. So most of the work to date that I've shown you so far in the presentation have been with the nCounter readout, but we have expanded the technology and are developing and will be commercializing the library prep reagents and all the bioinformatics to bring that data back in to enable you to go take that microtiter plate, make sequencing libraries, sequence it, and then analyze that data. And this just shows one example here where you're looking at different regions within a sample. If you do bulk analysis, and you rank those by the highest expressors to lowest expressors shown here, and then you do regional analysis, you see, if you keep that ranking the same from highest to lowest, that you see quite a bit of heterogeneity in the expression. So they don't oftentimes look very much like the bulk analysis, which just highlights the value of doing spatial analysis at this high-plex RNA level. Again, our initial panel that we are developing and will be commercializing next year is an aggregation of our 3 gold standard off-the-shelf panels. So these are really our best-selling panels, our most popular panels for cancer analysis. And it combines the cancer pathway, which really brings in a lot of content around the tumor itself, the pan-cancer immune profiling, where we have a lot more immune-related content, and then the pan-cancer IO360 assay, which is much more focused on targetable pathways. And so with that, you're getting a lot of spatial information about the immune system, the tumor itself, the microenvironment, and then we have quite a few important positive and negative controls within that allow you to analyze and normalize the data across the different regions of interest and across the different samples. Another look at what that allows you to do is it's enough content that you can really start looking at different pathways. So looking at, for example, these different regions of interest here selected along the invasive margin. We have one invasive margin here, so blue is essentially cold for these pathways. These are actually interactomes, so interactions. So cold versus hot in the brighter colors. And you can see these 2 invasive margins have very different biology. So one is a very inactive invasive margin, while one is a very active invasive margin. So all of this is wrapped up into a really well-designed interactive software. So the software within the system allows you to, in what we call the control center, allows you to do this, setting up the scanning, doing the scanning of all the samples, selecting your regions of interest, doing all that segmentation into tumor and stroma and so forth, and then collecting those tags into the plate. Once that plate of release tags goes out, either for nCounter analysis or ultimately for sequencing analysis as well, that data then comes back into the same software, and you go into sort of the other component of it, which is the GeoMx data analysis. And that allows you to have access to all of your images, all your ROIs, all your different probes, all your workflow across the top, which is QC, background subtraction, normalization, differential analysis, and ultimately visualization of that data in clusters and volcano plots and scatter plots and bar charts and all sorts of different ways of looking at the data depending on what you're asking. So this is all integrated. Importantly, you can export images, you can export data at any time if you want to take that data, either the raw counts, or any sort of normalized data, and you can go into R or Excel or your favorite statistical analysis tool. So it provides a great suite of tools that can get you right from sample to publication quality results and images, but there's clearly biostatisticians that want to interrogate the data independently of the software, and that's perfectly fine. You're able to bring in other sets of data, sample annotations. That's a really critical part. So you're able to go in and annotate all those samples to incorporate any sort of clinical data or metadata that you have associated with it. You can then feed that into the analysis protocols and then a variety of other data as well. And another component of the software is how interactive it is so that you can work as a team of scientists, as collaborators to run these studies. So you have the piece at the bench, which is loading the samples, setting up the scans. Once those scans have been set up, you can do ROI selection, you can do ROI approval from another site. So it's sort of a local cloud type infrastructure. The software all runs in the system, but different people can access it at the same time. So this person's scanning, this person's doing ROI selection from their own computer and initiating the collection. Once the data is generated and brought back in, you can analyze the data, you can collaborate with others, biostatisticians, for example. You can all be looking at the data and analyzing the data at the same time that the technician is back here running the next set of samples. So you can have multiple people interacting with their data at the same time. And importantly, it's all controlled through groups and so forth so that you don't have sort of the wrong people looking at the wrong data. We don't aim to be everything to everyone, so we have set up collaborations and partnerships with best-in-class companies in a variety of spaces related to this technology. On the top is Leica, so we have automated workflows for both the RNA prep and the protein prep using the BondRx system. With ACD, so these are the RNA scope probes, we're able to highlight and do ROI selection and look at the morphology of the tissue based on the RNA expression in situ. So that's using the ACD RNA scope probes. And then AbCam, which has over 6,000 IHC validated antibodies, and that allows you sort of easier, faster access to customize your panels as you see fit. The AbCam antibodies is a great source, but again, if you have another antibody that's not from AbCam, that's fine. We would just put a custom barcode on that, and then you can spike that into the panel to expand it. So just as a summary, the GeoMx platform, it's multiplex, so you can look at many analytes on one tissue slice in a single pass. It's multi-analyte. You can do both RNA and protein analysis spatially with this platform. It's simple counts, so it's a very quantitative assay, very large dynamic range, and the software sort of brings all that imaging and count data together into one view. You can shine those UV lights down to features as small as single cells. So there's a lot of flexibility in how you define your regions of interest and how you segment those regions of interest. One thing I didn't mention it is non-destructive. So you've done IHC or you've done in situ hybridization. Once it goes into the system, you're just shining that UV light and releasing the tags. When you're done with that, you can mount the slide. It can then be used for other applications. And the throughput is about 12 samples per day. So when you think about relatively large studies, you can churn through quite a few samples from a clinical, a retrospective clinical study, for example, on the platform. So it's got excellent throughput for those applications. Thank you. And with that, I'm happy to take any questions.
Great. Thank you, Dr. Hinerfeld. We did have a few questions come in, so we'll just jump right in. The first question is, can you use custom visualization markers?
Yeah, you can. So we provide off-the-shelf visualization markers. For example, in the protein assays, we have CD45 and pancytokeratin in a DNA dye. For melanoma, we substitute the pancytokeratin for a melanocyte-specific marker. We leave open, it's a 4-channel fluorescent microscope, so immediately we leave open one channel for your custom marker, and it could be, for example, CD68 for macrophages. That said, we provide you with all the information on the fluorophores that we use, and you can design completely custom morphology markers. Same is true on the RNA scope side. For RNA, you have a lot of flexibility, so it's entirely customizable.
The next question we have is, does the GeoMx work on frozen samples?
It does, yep. So we have protocols for frozen sample analysis for both protein and RNA. So while the main focus in terms of the validation of our antibodies and the building of the content has been on FFPE samples, we've cross-tested this on frozen samples and provide workflows for that as well.
Great. Another we received is, what is the maximum plex that can be analyzed on the GeoMx?
Yeah, so we have, we're developing this 1500 gene panel. It's about 1500 genes for using the high throughput sequencing readout. We've done actually some really interesting studies where as we were building out those larger panels to get to 1500 genes, we took an initial data set of a few hundred genes. So if you analyze the same sample with those few hundred genes, and then you analyze it with the 1500 genes, those few hundred genes are embedded in the 1500 genes, and compare the data has a very high correlation. So what that tells us is that as you continue to add more probes for more genes, there doesn't seem to be much interference. And in fact, we've done that, I believe, up to around 15,000 probes included. So from what we can tell, it's highly expandable to doing very, very large numbers of genes. And the sequencing gives us that room for the sort of counting or analysis of all those probes. So we haven't quite found the limit yet, but it's quite high.
Looks like in the interest of time, we'll have to stop here today. Once again, I'd like to thank Dr. Douglas Hinerfeld for his presentation and all the attendees for joining us. If you'd like to watch the presentation again, or any of the other webinars that are a part of Webinar Week, the recordings will be available at genewiz.com/webinar-week.